We have some friends from Kuwait and Virginia in the United States, Alaska. Welcome everyone. Bhutan, Australia, Jordan. So nice to see everyone from around the world. Latvia. Australia, Alabama. Hi, Elena. Glad to see you here. Looks like some old friends and new friends joining us today. For those of you who don't know me, I'm Sarah Saladay. You might have known me as Sarah Silverberg years ago now. I'm coming to you today from the University of New Hampshire in the United States. We're in the northeastern part of the country, and I'm really far north. I'm about an hour from the Canadian border in New Hampshire. It's a bit rainy here today, and I'm happy to be here. Peggy, take it away.
Hi, I'm Peggy Valletta from California, near Monterey Bay, Elkhorn Slough National Estuarine Research Reserve, where I am the partnership coordinator. Hi to everyone.
Hi, I'm Mike Chabot from the State University of New York at Fredonia, about an hour south of Buffalo, so far southwestern New York State. Finally today, it's not raining. It's been a little bit like being in the tropics, and really excited to be here and learn from all of you.
So we just want to get started today by a little housekeeping. So first of all, I just want to remind everyone that this is not a protocol training, but we will be highlighting several parts of the carbon cycle field protocols modules today. Of course, you can check out the full e-training on the Globe website, and you can become trained by participating after this workshop. During the workshop, we'll have about an hour together, and you'll have many opportunities to participate. The first is your questions. So you can always ask a question in the chat, and Peggy and Mike will do their best to answer them. And then I'm going to provide another link momentarily to enter questions in, where we'll be able to kind of log those questions and get to them in the protocol question and answer session tomorrow. So just hold on and I'll put up that link. Or rather, Peggy will probably put up that link. And there's going to be lots of links to interactive components during our workshop today. So it'll come up in the chat, and then you will copy and paste it into your browser window. And then you'll do the activity, and then you'll come back to the Zoom window so we can talk about it, and I'll show it on our shared screen. So as we go along, don't hesitate to let us know what you need. If I need to slow down a little bit, please mention something. I do speak a little fast sometimes, but yesterday I did all right. And this workshop is being recorded, and you'll be able to access it at another time if you missed something. So here's Mentimeter. Mentimeter is a program that we're using today to capture your questions so that we can answer them later. So if you have questions about the carbon cycle introduction module, if you were able to complete that, this would be a great place to ask some of those questions. And also any questions that you come up with after the end of the session today, you can put those into Mentimeter, and we'll make sure to answer those tomorrow or Thursday during the question and answer session. So why are you all here? Why should we even collect carbon cycle data? As most of you are aware, the carbon cycle is a main driver in climate change, and scientists collect the data in order to understand how terrestrial ecosystems will respond to warmer temperatures and higher CO2 in the atmosphere. And in particular, carbon data that you're collecting on the ground with teachers and students can contribute to a better understanding of the relationship between carbon storage and surface climate. And that'll become more obvious as we go through today. So like all good trainers and teachers, we're going to frame our whole workshop today around a research question. So today's research question is, how do differences in tree species and sample site affect carbon storage near where I live? So how do differences in tree species and sample site type affect carbon storage near where I live? We have other examples of questions that you can ask in our protocol modules. And there's also some great student research projects that have asked similar questions. So in order to answer our question today, we're going to start with something called a KWL. Many of you have probably seen this or used this before, but it's a table where you enter what you know and what you want to know about the subject. And then at the end of the learning, you write down what you've learned, kind of keep track in your brain, sort of where your thinking is at. So in the Google chat, there is a link and you can go to that Jamboard and you're going to be looking at frame 1. So copy and paste that link into your browser window. And you should see something like this. I see lots of people logging in and already starting their work. That's great. If you're not quite sure how to start, you just click on this little sticky note button over here and start putting in your ideas. So remember, we are focused in particular on our question, how do differences in tree species and sample site type affect carbon storage near where I live? We will talk about a few things more generally, but that's the question we're really focused on today. I love the things coming up in the no column. Looks like some people did participate in the carbon cycle introductory module. I'm 50% carbon after squeezing out all the water. Carbon is crucial for life. Carbon is stored in all living things. Net primary productivity creates biomass. What are some more things that we think we want to know in order to be able to answer today's question? What do you need to know about? So maybe one more moment, type up your last thought here and get it into the KWL. So I see a lot of questions here in the what I want to know that we're not going to go over today, mostly for time's sake, but they are great questions. I'll try to get to some of them as they relate to today's research question. But just as a note, the carbon cycle materials were developed focused on forest ecosystems and then spread out to other terrestrial ecosystems, both native and non-native. We did not do any activities specifically focused on soils or water bodies. There is information about that out there, but we did not develop any protocols for that. We also focus on the science of the vegetation. So we do not do anything with carbon footprints, but there are other activities available through, I think, even NASA and some of the other sites that'll have you go through your carbon footprint. So in terms of today, how do differences in tree species and sample site type affect carbon storage near where I live? So we'll definitely want to know how to identify trees, how carbon storage varies with forest type. Are evergreens better than deciduous? We'll definitely cover some of these today. Do all trees absorb carbon dioxide? We won't directly address the impact of deforestation, but I think you'll see how you can think about that after you've actually measured carbon in trees. Which trees absorb the most CO2? We'll get at that concept. How to determine tree storage by type. Where carbon goes after a tree decomposes. That's dealt with a little bit more at the global level. If you read the carbon cycle adventure story. How to get underground carbon storage. Again, we're not going to deal with that too much, but it is dealt with a little bit in some of the data analysis. Old trees versus young trees, that'll sort of generally come up. Yep. Okay, so back to the next part of our program here. So what are we kind of dealing with today? Hopefully you had a chance to look at the introductory activities. If you didn't, that module is available and will answer some of those questions that you have. Field learning activities, we're going to take a look at allometry and biomass units today. We're going to also talk about site selection. We're not going to do site setup because we're not live together. And we'll talk a little bit about the 3 different protocols here for trees, shrub and sapling, and herbaceous, but we're not going to dig into it because those protocol sheets are written up and they're pretty clear. We're not going to do data entry into the Globe database today, but we are going to take a look at the data analysis and what's happening in the background on the Globe website so you have a better understanding of how carbon is actually being calculated so that you can describe that when you're teaching or training. So answering our research questions, we're really going to focus on the protocol of site setup, learning activity biomass units, part 2, learning activity allometry, not allometry, part 2, and a supporting protocol, which is the data analysis spreadsheet. So how do you even get started? So remember our question deals with trees specifically today. So the first thing you would do is go out and measure a tree. What is the tree's circumference? If anyone's participated in land cover protocols, it's the same tree circumference measurements. And then of course, there is a relationship between circumference and diameter. And we have an activity to do that work and help students understand that work, but we're not going to cover it today because most adults already understand this relationship of a circle. To get from diameter at breast height to biomass, there are special equations. We'll talk about those today. To get from biomass to carbon storage, first we have to know what biomass is, and then we have to know something about multiplying by 50% and why that is. And this is all for a single tree along this line until right here. We'll talk a little bit about how to use your sample site area in order to scale up and get carbon storage for a whole sample site. So we're going to start here at the beginning and talk about actually going out to set up your sample site. I just noticed that Jody Haney, who has worked with us to do some of these protocols before, has a great footprint calculator there in the chat. You can record that link for another time if you like. So site selection, how do you select a site? So the first thing that you're going to do is decide whether your site is a standard or non-standard site. So we're going to do some activities to make that a little bit clearer. So first of all, your standard site is an accessible area of at least 225 square meters. It could be a 15 by 15 meter square, but it could be also a different shape like these show, whatever kind of works for your area. It could be as large as 30 by 30 meters. If you already have a land cover site, you can use the same site setup for that. And it's contiguous vegetation. So contiguous meaning there's no breaks in the vegetation. There's no sidewalks, there's no buildings, there's nothing interrupting the natural patterns of the vegetation there. And that's versus a non-standard site, an accessible area of 225 square meters again, but it's going to have some vegetation that is growing or perhaps planted, so it could be natural or planted vegetation, and then some level of human interference. So Peggy's put up the link in the chat, and you can go to that link and practice for yourself a little bit. What defines a standard site versus a non-standard site? So you can click on the boxes and drag them around to where you think they go and make it clear to your own mind, what do the standard sites look like versus the non-standard sites? So you can take a few minutes to do that now. Remember, if you have questions coming up, Mike and Peggy are happy to answer questions in the chat. So you can save your flippity for later. I'm going to click over to a flippity that is completed, so we can take a look at what it looks like here. Oh, or not. Ha. Thought I had already done it. But you can do it along with me if you're still confused. Get it sorted quick here. If you'll notice, what's coming up in the non-standard site are all of our areas that look like suburban and urban settings. Our standard sites, we have some natural ecosystems. Both locations have at least 225 square meters. Both of them are accessible for your training and your teaching. You don't want to have to go super far every time. And then I've got this picture here. You might notice that it's a lot of trees standing in rows. This is a tree plantation. And because it's contiguous vegetation, we would call this a standard site. It's not interrupted by human interference in terms of man-made structures. Even though humans planted the trees here, there's no man-made structures that would affect the carbon storage per meter squared. Because that's really the difference here between the standard and the non-standard site is when you go to divide your vegetation by the whole area, are you looking at kind of this complete coverage of vegetation across your site, or is the vegetation interrupted by all of these other features? So it drastically changes your grams per meter squared of biomass. We wanted you to note that teachers could set up both types of sites if they have sites close enough to each other. Perhaps they do a schoolyard site like this one out in front of their schoolyard, or a city block with a school on it. And then they go on a field trip for 2 days, or they go on an overnight trip, and then they do a forest site out on their field trip that they maybe only visit once or twice a year. So really think about how these two different kinds of sites might impact carbon storage near where you live. So now what I want you to do, you're gonna open up another tab. And you're going to show us and describe to us what your sample site is that you would use to train or teach these carbon cycle materials. So you're thinking now about your own location, and you're planning to do a training, or you're planning to have a group of students, and what does your site look like? And do you think it is a standard site or a non-standard site, or you really don't know and you need help? So after you've posted yours, you're going to be able to give feedback to other people. So when you put the link from Padlet in, this is what you'll see. If you know it's standard, put it here in the first column. If you know it's non-standard, put it in the second column. And if you're really not sure, put it in the third column, and we can add comments. So take a few moments now. Another way to think about standard and non-standard is a standard site would be representative of the biome that you live in, while a non-standard site might be just representative of your local area, like the city that you live in. Good, I see we've got some local parks. The riparian zone right next to a river. Conifer forest. Good. I see that a couple people have 2 sites that are nearby to them that they could use. The playground with dispersed trees is a perfect location for a non-standard site. My riparian zone friend here has one social path where people walk. This is a great question. Would this count? So if you can set your site just off the trail, so the trail wouldn't be included and impact the actual amount of vegetation that you're measuring, you could use it as a non-standard site. And I'll show you a picture of our site that we're going to talk about today as an example of that, to answer your question. Great. I'm going to let people continue to enter their thoughts here, and Mike will monitor and add any comments if people have more questions. We'll go back to our presentation screen. So the site we're going to do some analysis on today is the College Woods Natural Area, and it's a 64-acre tract of land next to the university that was set aside for enjoyment as well as research study. So here's a side view in the middle of the forest. You can see there are tall, mostly evergreen trees with a path and very little shrub, sapling, or understory growth. And then in this satellite image, you can see this dotted line. That's that same path. So when we set up our field plot for training teachers, we put it here next to the path so that we wouldn't have to include the path in our total area for measurement. Yes, contiguous vegetation is a standard site, whether it's native or non-native or invasive species. As long as it's all vegetated with no human-made structures taking up the area. So once you've selected your area and you've set out the boundaries of your site, you're going to go through some protocol decision making. So this is a decision tree that's available for both standard and non-standard sites. So this is for a standard site. But you always start at the top and read the first question. Do you have trees greater than 15 centimeters circumference on your sample site? So think about the site we're doing today. Our answer is yes. Then we follow that down to the next question. Do you have shrubs and saplings on greater than 25% of your sample site? Greater than 25% means they're actually going to have a big impact on carbon storage on your site. If there are less than if they fill up less than 25% of your site, they're not going to have enough impact on carbon storage that you need to measure them, because trees just store so much more carbon than smaller forms of vegetation. So in this case, we're going to say no, because we're looking at college woods in our example. And then, do you have herbaceous vegetation on greater than 50% of your sample site? And again, that has to do with how much carbon is stored in grasses versus stored in trees. So we did not have herbaceous vegetation on greater than 50%, so we say no. So today we're just going to focus for our example on trees, but each one of these separate questions does in fact relate to an individual protocol that has a protocol sheet and data sheets associated with it. And they're just like all the rest of the globe sheets that you can very clearly follow through. So how do we know what to do next? Well, we have to have some concept of what biomass actually is in order to be able to move forward. So biomass is the total mass of all living things in a given area, which is why on a non-standard site, we have to be careful because there's a lot of things that are not biomass, like a building. And when we calculate, once we can measure biomass, we can calculate carbon because carbon is 50% of biomass by dry mass. So if you imagine a square meter of grass and you cut it down all the way, and then you carry that grass to your oven and you put it in and you dry it out and you take it out and you put it on the scale, that is your dry mass. That's your dry mass, and then multiply by 50%, and that's carbon. So someone said that people are 50% carbon if you like wrung us out like a sponge. So you got to remove the water first, and then 50% of that is carbon. And that's true for all living things on planet Earth, you know, plus or minus a percentage point. Another thing to note about biomass is that vegetative biomass is so great on the global scale that we, as scientists, when we talk about the global carbon cycle, we measure vegetative biomass because it's really, really important. But on the global scale, you actually don't see animal biomass measured anywhere because by comparison, it's so much smaller than vegetation, with the exception of here in the soils. Soils is a separate number calculated by scientists, and the microorganisms that make up soil are actually a huge component of the soil carbon storage. So above ground and below ground carbon is quite different. And then another thing that's really important to note about biomass is that getting an understanding of local plant communities and the amount of biomass that they store provides really important information about carbon storage on those micro scales. So measuring with students actually can be really important for scientists in your local area, depending on the work that scientists in your area are doing. So carbon cycle learning activity Biomass Units has two main parts. The first part is sort of focused on students and sort of getting at the conceptual idea of what is biomass, what are the units of biomass, which is grams per meter squared, so mass per area, and it has them doing it for themselves. So you take the mass of all the students in the classroom, and then you take the size of the classroom, and then and you can get biomass in grams per meter squared. And then you can play around with that idea. So what would happen if you put the same students in a gym, a big gymnasium? What would happen if you put the same students inside a closet? How would the biomass change? Or likewise, if you had, the next door classroom join all of your students in the same classroom, how does the biomass change then? So these are important concepts for students to understand because it relates back to the vegetation, because biomes have different amounts of biomass and carbon storage. So part 2 really focuses on the vegetation, and we're going to do a little bit of work with that in just a moment. So in part 2 of the activity, students rank global biomes from greatest to least biomass, and then compare their guess to available data that's been collected by scientists. And then they have an opportunity to estimate how much carbon is stored in their own schoolyard based on their biome classification and what they can observe. So if you're doing a non-standard site, you might start with your biome, but then you'll have to think about what else is on your site aside from the vegetation. So we're going to do an activity on Jamboard again. So you can go back to that original Jamboard. Peggy will put the link in again just to be sure, but if you go back to your original Jamboard, and at the top, there's going to be some different frames, and you're going to click over to the second frame, and you'll find a global biome map. This was adapted from the World Wildlife Fund. And you're going to use your drawing tool here on the left to mark where you think the most plant biomass is in the world. Which biomes have the highest plant biomass? And the key is down at the bottom. You made a mistake. There's also a little eraser here. Second one down, you can erase your work. You need to. I see someone did circle the ocean, perhaps. If that was a mistake, that's no problem, but we're not dealing with water bodies in this particular carbon cycle study, just land biomes. And you see lots of people focused on our tropical and subtropical moist broadleaf forests. A few people have circled our tropical and subtropical grasslands and savannas. People are noticing that there are some similar biomes around the world. Oh, someone has guessed that the temperate conifer forest has the highest biome. Once you've done the highest, you can click over to the third frame and do the lowest biomass. People jumping right in. Some guesses that the rock and ice areas of the world have the lowest biomass. And others guessing that deserts and xeric shrublands have the lowest biomass. Montane grasslands and shrublands. The Nepal Everest region. So let's take a look at what the scientists have gathered. So if you notice that here it says ecosystem types, so different groups of scientists classified biomes and ecosystems slightly differently, but you'll get the main idea here. And in fact, tropical rainforests, or those moist broadleaf forests, do have the most biomass in the world. In grams per meter squared, they have 40,000 grams per meter squared of biomass. And then way up at the top is our least biomass in the rock, ice, and sand categories. A lot of people picked up on that this time. And that's compared to the tundra and alpine meadow that have 600, as well as the desert scrub. So deserts and tundras actually do have more plant biomass than we perceive them to have. But when compared with tropical rainforests, it's hardly a comparison in terms of biomass and therefore carbon storage. So I want you to consider what students might consider for a moment and estimate how much carbon might be stored in the sample site area you described earlier for your own area. So think about your own place that you live. And you might just note this down for when you actually do your training and your measurements later. Think for yourself, your own area of the world. So now I want you to see how well you're doing with this thought exercise, and think about what would be a good biomass estimate for today's example. College Woods in the northeastern United States, and type your estimate into the chat. So what's a good biomass estimate for today's example in College Woods, in the northeastern United States? So you can always go back to that Jamboard and see what biome we're part of. And then look here. Just a reminder, this is the College Woods site we were looking at. How much biomass do you think it has? Okay, so keep that biomass guess in your head, because we're going to actually look at that number later. We're going to look at the calculations made by our spreadsheet. But moving on for now, how do we actually go from our measurement of tree circumference, or DBH, to biomass? And we have to use something called allometry, or allometric equations. So what is allometry? How do you measure the dry mass of living plants in a given area without chopping them down, drying them, and weighing them? We're not going to go chop down our trees like we chopped down our grass earlier in my example. But scientists and foresters and even loggers kind of participated in this task over the last 100 years or so. And so this data has been collected over time, and scientists have taken time to figure out the relationship. So allometry at its very core is the relationship between the part of an organism and the whole organism. And for trees, scientists have been able to figure out that there's a consistent relationship between the diameter at breast height, 1.35 meters off the ground, and the whole biomass of the tree. And they can actually even include roots in that estimate, but it depends on whether or not the roots were actually measured initially when they cut down the trees, like if they pulled all the stumps out and added that in, or whether or not the equation they created was just for the above ground biomass. And this exact relationship between DBH and biomass does vary with tree species group. I know some people thought that we needed to know about that, so here's where we're going to talk about that. And those differences are based on both the cell layout of the individual tree species, as well as the growth rates of those species. And both those things together contribute to wood density. So wood density is really the driver here of how different tree species store biomass. So we'll come back to that concept in one moment. In our allometry activity, first we have to make sure that students understand that you can actually calculate relationships between parts of an organism and their whole. So just like in biomass, there's a concrete example, so students can measure their height, arm span, and foot length, and show how those different parts are related by creating linear equations. So it's a great math connection as well. And then once they understand how those really work, they get back to this vegetation component. So students use the concept to understand how circumference and DBH of trees can be used to estimate biomass. And to do that, they're really viewing the allometric relationships of tree species groups. So the graph on the right is called the predicted biomass graph, and it was created by Jennifer Jenkins and her team. She's a US forester And they did a meta-analysis of 100 different studies that developed allometric relationships for trees across the United States. And she noted that this compilation of equations was needed in order to better understand biomass and carbon storage on a national scale. But she made a really important note, actually, that's at the beginning of the paper, that other countries in the world have completed similar national assessments. So if you're an international participant with us today and you're saying to yourself, well, these aren't my species groups, do know that this concept exists globally and that it is very likely that there are scientific papers published in your region for your tree types. So you would want to talk to your local universities, local forest service, whoever manages forests in your country to find out if they have something like this. So what exactly are we looking at? The graph on the right shows diameter at breast height along the x-axis from 0 to 50 centimeters, and then predicted biomass in kilograms from 0 to 1600 on the y-axis. And then each individual line represents a single species group. And that group of species is pulled together because of their density, right? So the number 9, the cedar larch group. So all of the species in the cedar larch species group have similar densities and therefore behave similarly. When you measure their DBH, they have a predicted amount of biomass. But that predicted amount of biomass is different than, say, species group 1, maple, oak, hickory, beech. So it's a bit of a thought exercise here. Take a look at maple, oak, hickory, beech, group 1. And if we measured a tree that was 30 centimeters diameter at breast height, what would the predicted biomass be for group 1? You can put that in the chat now. So DBH, if we measured a tree that was let's say an oak tree in species group 1, 30 centimeters, how much would we predict its biomass to be in kilograms? See a few guesses coming in: 600, 560, 500. So do you know how to use this graph? Can you figure out if a tree were 30 centimeters diameter and it were in species group 1, what is its predicted biomass? We follow it across here. Good. People trying to interpret these lines, these little tiny lines. Looks like somewhere around 600. Most people are guessing just slightly less. So 30 centimeters diameter, you kind of draw a line straight up and then across at a 90-degree angle to try to find the predicted biomass. So if your students or teachers are unfamiliar with density, the end of the allometry activity does have an extension that you can follow through on, where you actually take wood blocks of different wood types and do a bit of a density experiment. So how does this all relate back to the data that we're actually going to collect? Remember, we're measuring tree circumference, and we've measured our sample site, and everything else is a calculation. So we're going to take a moment to look at those calculations in a spreadsheet. So this spreadsheet is available on the Globe website. It's a template. You don't need to go to this link now, but I wanted to provide it to you so that you can go back and look later. and play with it yourself. And this is what it looks like. So normally when you enter your data into the Globe website, your data goes through a black box and essentially spits out carbon storage for your plot. You put in your tree circumference and you put in your sample site area, and everything else is magic to you. Here, we're going to show you that it's not magic. So this page is your instructions. And here we are, plot size. I've entered my 30 by 30 meter plot. And then I click on field data entry, tab 3 along the bottom. And here's all the data that I actually went out and measured with my team. So tree number southeast quadrant number 13 was a fir hemlock, and it was 15 centimeters circumference. So how did I get this species group? If I skip along the bottom, I can get to tab 8. This is a handy little function that helps you find species groups quickly. So for hemlock, let's say I wanted to find out where a specific tree was found. So I could do a find function, Control F, and I might say, "Ooh, I know I have a pine." Oh, look at that. I have 2 out of 74, and there's actually a whole pine species group. So even if I don't know what kind of pine is, I know it falls into this species group only. But let's say I'm from another country, and here's the data that you all collected. And Acer platanoides, that's a US tree, but here we have Acer rubra from Argentina. So we can plug in Acer here. And we find out that a number of our Acer species, our maples, are in the maple oak category. But we also have a bunch of Acers in the soft maple birch category. But here, in fact, there's an Acer rubrum. And so that's in a different category. So you can check here. But of course, if we enter a tree from Taiwan or Thailand, very likely it won't be in this sheet, and you'd have to talk to your local forester. So once we know that, we can check out what's actually happening. So for each tree species group, there is a specific relationship, as we pointed out, and it's always that biomass equals the exponential function of the coefficient B0 plus the coefficient of B1 times the log of DBH. And so here, built into the spreadsheet, are those coefficients that create these lines. These curves are all created by that single equation here with the coefficients plugged in. So that's important to know. And so people also had a question about roots. So in this spreadsheet, you can also investigate components of the tree: foliage, stem wood, bark, and roots. And there's an interesting graph here that you can investigate further about the size of the tree and the components and how they change biomass and predicted carbon by ratio. So once you have that information, you can do a test for yourself if you'd like. There's an equations tab. So sometimes Excel is sort of complicated for people and they don't really understand it. But in the equations tab, we've built in how the equation looks in Excel and showed you how you can do it on a calculator. So even if you only do it for one tree, you can check to see that it works. So here's from my database in the spreadsheet. And here's the way I would do it on my calculator. And on my calculator, I got 345 kilograms of carbon, or kilograms of biomass. And here, using the spreadsheet, You'll see the equation up here in the formula bar. And we get the same answer. So that's just important for you to know in terms of working with the data yourself. And now we can go back. So we had that field data. We can go back to tab 4. We understand how everything works. And here it is. This is the money, people. If you weren't listening before, listen now. The data you entered is here in blue. The coefficients that we just saw on that other page are in blue on this side. And in the middle are our calculations for each individual tree, following that same flow chart that we followed the whole presentation. So circumference of 15 gives us a diameter of 4.7. And then you can see up here in the formula bar, we just divided by pi. Here, total above ground biomass in kilograms. Here it is, that equation, the coefficients plus the natural log. And finally, we convert to grams and then to carbon by multiplying by 50%. This is carbon for this one individual tree. And then if we add up all the trees, we come up here to our summary data. So plot biomass was 31 million grams per plot. So that's in comparison to that petagram number, right? You could figure that out. And then this was kind of, these two are the most important for us because they're already taken for your area. They've taken account of your area in grams per meter squared. So what did people guess for College Woods was going to be the biomass? They guessed, I think, around 27,000 grams were a lot of the guesses. And we actually have 34,935 grams per meter squared of carbon in the College Woods plot. So kind of making that connection between biomass units activity, your estimate, and then the actual biomass on the ground and carbon on the ground. So we're almost ready to wrap it up here. My team's telling me I'm just about out of time, but I want you to take the last moments here to do something really important, which is synthesize your learning. So you're going to head back to the Jamboard, back to frame 1. Frame 1 of your Jamboard was that KWL. And type in what you have learned about our question. And we're going to leave this Jamboard up for the rest of the day. So you can feel free to continue to add your sticky notes and screenshot it at the end of the day so that you have your thoughts and everyone else's as a good example of how to do this in a training or with students. There we go. That's what it is. Good. Somebody's done some relearning about things they had forgotten. My screen is clearly not working. People are adding new frames. You don't want to add new frames here. You just want to go back to frame 1. Back to frame 1. Oh, did someone accidentally clear a frame? Oh no.
Yeah, I think frame 1 got cleared. Yeah.
Okay. Certainly you could still put your what you learned in frame one, people are adapting. Great. Perfect. Good. More clarification on site selection difference. That was one thing we hoped to accomplish. Calculating biomass. What else did you learn today? As you're doing this, I'll just let you know that there is a student research project that Peggy has linked in the chat that you can open up in a separate window and check out on your own time. But it's a student-driven project that was actually presented at an SRS in 2019. So you can see real students using their work to understand carbon in their own backyards. So again, keep adding here so people can see what we've learned about our question. How did differences in tree species and sample site type affect carbon storage near where I live? And you can keep adding questions to the Mentimeter that you got at the beginning. And we'll try to answer those questions the best we can on Thursday during the Q&A. It was so nice to see some of you again and see your names and thoughts in the chat. Thank you all for coming.
Thanks so much, Sarah. So we do not have any other scheduled sessions for today, but there are other resources that you can use. The recordings from the protocol sessions yesterday are up, along with the working group report out if you haven't watched that quite yet. So with the rest of your time until the sessions that start tomorrow, feel free to check out some of those resources. Thanks again, Sarah, for a wonderful training.
Hi, everyone. Nice to see you.